after the hypersensitivity types, we study autoimmune diseases. So, immune reactions against self-antigens is autoimmunity. Auto means self. So the immune reaction against self-antigens. These are important cause of certain diseases in humans. So this table gives some examples of autoimmune diseases. We saw in different types of hypersensitivity reactions that autoimmune diseases can be type 3, type 2, or type 4. So they can be of different types of hypersensitivity reactions. Autoimmune hemolytic anemia, autoimmune thrombocytopenia, myasthenia gravis, Graves' disease, for example, myasthenia gravis and Graves are type 2 hypersensitivity reaction. They are autoimmune diseases. Multiple sclerosis is another type of hypersensitivity reaction, is type 4, but again is an autoimmune disease. So autoimmune diseases have different mechanisms of development. So this table gives examples of autoimmune diseases. And autoimmune diseases can be organ-specific or systemic. Systemic means they involve many systems of our body. For example, systemic lupus erythematosus, it causes diffuse problems in many systems. Or rheumatoid arthritis. And some diseases are proved to be autoimmune, such as polyarteritis nodosa is a type of vasculitis, or IBD, including Crohn's disease and ulcerative colitis, or primary biliary cirrhosis. And some autoimmune diseases are organ-specific. For example, myasthenia gravis causes weakness of skeletal muscles, so it's an organ-specific, not systemic. Or Graves' disease causes hyperthyroidism. Goodpasture syndrome causes problems in lung and kidney. Or type 1 diabetes mellitus, which is the pathology in pancreas. So, autoimmune diseases are organ-specific or systemic with different examples. Now, if we want to study autoimmune diseases, first we should understand the concept of immunologic tolerance. So again, this is not pathology, this is immunology, that any healthy person should have immunologic tolerance or self-tolerance, not to let invaders, not to let self-antigens to cause pathology or cell injury in our body. So what is tolerance? What is immunology tolerance? Is the phenomenon of unresponsiveness to an antigen. So we shouldn't have any response to our own antigens, or else our healthy cells will be injured. So self-tolerance is present in any healthy person. And self-tolerance means unresponsiveness to self-antigens. Now, tolerance is divided into central tolerance and peripheral tolerance. As you know, lymphoid tissues are divided into two types, central and peripheral. Central include bone marrow and thymus, and peripheral or secondary include lymph nodes, spleen, and mucosal lymphoid tissues scattered in different organs such as gastrointestinal tract, lung. So, first, cells are selected in central lymphoid tissues. As you know, bone marrow is the site where B cells become mature. they are developed and become mature, and thymus is the place where T cells become mature. So central tolerance occurs in thymus and bone marrow. Central tolerance, so it means unresponsiveness to self-antigens. It means that if in thymus and bone marrow some B cells or T cells are hyperreactive against our antigens, our self-antigens, they should be deleted, or else they will cause tissue injury, they will cause cell injury, and its name is autoimmune disease. So it has some mechanisms. The first that is common in both thymus and bone marrow is apoptosis. So you see in this picture that this is thymus, this is bone marrow. So T cells in thymus and B cells in bone marrow, if they are hyperreactive to self-antigens, so this is an antigen presenting cell, for example macrophage or dendritic cell, it has processed a self-antigen and has brought it through MHC molecule to its surface. If it is a self-antigen, so the T cells shouldn't have any response to, any pathologic response, any immune response to this antigen. So if we want to have unresponsiveness, these hyperreactive T cells should be deleted by apoptosis. So tolerance or self-tolerance it includes central and peripheral tolerance. Central tolerance includes thymus and bone marrow. So in thymus, T cells and in bone marrow, B cells are selected. As you know, T cells have a receptor called T cell receptor. So some T cells occur in thymus. This is a T cell that has a T cell receptor TCR. Some T cells are hyperreactive against our own antigens. These antigens, as you know, are presented by MHC that are present on antigen-presenting cells. So some T cells that are hyperreactive against our self-antigens, these should be deleted by deletion by what? Apoptosis. And some B cells that have B cell receptor, what is its another name? Antibody, which has this structure. Again, some B cells have hyperreactive BCR that these also should be deleted. And this deletion again is by apoptosis. So one important common way of central tolerance in bone marrow and thymus is apoptosis, deletion by apoptosis. It means that for deletion of these kinds of cells, some apoptotic proteins should be activated. If you remember from chapter of cell injury, we have apoptotic and anti-apoptotic proteins, BCR. So in thymus and bone marrow, pro-apoptotic proteins are activated to cause apoptosis of these abnormal T cells and B cells for having a healthy self-tolerance. So this is the first way of central selection. So this kind of selection is called negative selection. So it means that
some cells should be deleted. Some cells that if they start living, they will cause injurious immune responses, which will be called the autoimmune diseases. So they should be deleted. And this type of deletion is called negative selection. So they are killed. So this is deletion and this is apoptosis, deletion by apoptosis. What is the second way of self-tolerance? In thymus, if you remember, we have three types of T cells. T helper cells, cytotoxic T cells, and regulatory T cells. So in thymus, regulatory T cells are also developed, which are called also reg cells, reg T cells. These regulatory T cells inhibit CD4 and CD8 T cells. So the other mechanism of self-tolerance in thymus is development of regulatory T cells. These regulatory T cells are released in circulation, in peripheral lymphoid tissue. So they go to periphery, to peripherals. So regulatory T cells are also important in peripheral self-tolerance. But for B cells, the second way of tolerance is different. It's called receptor editing. So it's not apoptosis. Some cells are killed by apoptosis. But some cells that have a B cell receptor or antibody that is abnormally reactive to self-antigens. So in nucleus, the DNA has some genes for making antibodies. Any molecule has a gene or multiple genes. So for antibody, we have some genes, such as B, D, J, because they can produce different light and heavy chains and variable and constant parts of antibodies. So we have different types of genes. So their arrangement on chromosome is changed. So imagine we have 3 genes, for example, for development of an antibody, this. So in nucleus, there are 3 genes, for example, on a chromosome that these A, B, and C, one allele, this A, B, C, another allele, they express molecules for production of this BCR or B cell receptor. But if it produces a B cell receptor which is not normal, is hyperreactive to self-antigens, this arrangement is changed. ABC, its order is changed to, for example, ACB or BCA. So the arrangement on chromosome changes. By changing the arrangement of these genes, so the product changes to a weaker type. So a type of B cell receptor will be produced that will not react with self-antigens. So this concept is receptor editing. Receptor editing.
And B cells, it's more for cancer.
In cancer?
B cells and T cells.
No, we can't say that. Both of them, both antibody-mediated and cell-mediated, are important, and especially cell-mediated. If you remember CD8 cytotoxic T cells, they work against tumor cells and viral infections. But it depends on the tumor type. So any disease has its different type of mechanism of action. So Central tolerance in B cells and T cells include apoptosis. In thymus, it also includes development of regulatory T cells, and in bone marrow for B cell receptor editing. So this is for central cell tolerance. Now in periphery. In periphery, not all cells in thymus can be negatively selected. I mean, cannot be killed by apoptosis or inhibited by this inside the thymus. Some cells escape to circulation. So there should be also some mechanisms in peripheral lymphoid tissues to inhibit these types of T cells or B cells that have abnormally hyperreactive to self-antigens. Again, for T cells and B cells, we have mechanisms. For T cells, as you know, Treg is produced in thymus. They enter the peripheral lymphoid tissues. They can inhibit those hyperreactive T cells. So T regulatory cells are immune suppressors. They can inhibit these abnormal T cells. And the other way of tolerance in periphery is called anergy. Anergy is both for T cells and B cells. What is anergy? Is failure to respond. So anergy is a term for peripheral tolerance. Anergy. So anergy means no response to the antigen. Failure to respond. It's both for T cells and B cells. So the mechanism in peripheral tolerance is by T regulatory cells for T cells, and also anergy for T cells, and for B cells again anergy. As you know, for any immune response, this is an antigen presenting cell that brings an antigen by MHC molecule to a CD, a helper, a T cell generally, that has a T cell receptor. For having an immune response, this is the first signal, signal 1. But this is not enough for having an immune response. We need a second signal, again between this antigen presenting cell and T cell. Also, the same is for B cell. It's called second signal. So there is a marker on T cell called CD28, and a marker on antigen presenting cell called B7. Together with this first signal, if the second signal is also activated, then we will have immune response. But in any way, if these receptors are blocked, we won't have any immune response. And this is what happens in anergy.
Blood energy.
What?
Blood energy.
In anergy, so in normal immune system, anergy, it means not having the second signal. So by some molecules, these receptors are blocked. So immune response will not be present. This is failure of response in T cells and B cells. So anergy is a peripheral way of self-tolerance in both T cells and B cells. So this is what happens not to let development of autoimmune diseases. But in autoimmune diseases, these processes, any of these processes are disordered. There is an important protein in thymus for central tolerance. I give an example. In thymus, which is important for central tolerance, there is a protein called IRE, autoimmune regulatory protein, IRE. This is a protein that it should be present in thymus because it is very important for deletion of abnormal T cells. So some people, they have some mutated genes that this IRA is not expressed, and they develop a severe autoimmune disease. It's called polyendocrinopathy, that it involves many endocrine glands. So we learned an important protein in thymus called IRA, autoimmune regulatory protein, that it should be present for negative selection of hyperreactive T cells. In some people, this IRA is mutated, its gene is mutated, and they have autoimmune diseases. So if there is problem in any of these processes, for example, if you imagine the second signal cannot be inhibited automatically by our healthy immune system, so energy will not be present and the patient will have autoimmune disease. And some toxins and some drugs change our molecules, our receptors. So you can see that the mechanism of action of some infections and some molecules, toxins, can cause autoimmune diseases. That's why some of the causes of autoimmune disease are imagined to be infections. Some bacteria, some viruses. So this is central and peripheral tolerance for prevention of autoimmune diseases. Some genes are important in autoimmune disease. Maybe you can imagine that it is MHC or HLA. MHC genes or HLA, which is human leukocyte antigen, is the same as MHC. So HLA genes are important because they have many types in people, that some of them make people susceptible to autoimmune diseases. So genes are important in development of autoimmune diseases. One of them is HLA. So role of susceptibility gene, association of HLA alleles with disease. We have different types of HLA genes. We have a table that shows the relation of different types of HLA genes with different diseases. One of them that is proved and very strongly related is relation of HLA-B27 with ankylosing spondylitis. So other diseases are also mentioned here that they are related with different types of HLAs. Other genes can also be related with autoimmune diseases. As example, for example, PTPN22 is a gene that its mutation is related with rheumatoid arthritis, type 1 diabetes, and not like receptors in innate immunity. Polymorphisms in a gene for NOD2 is associated with Crohn's disease and inflammatory bowel disease. Or polymorphisms in genes encoding interleukin-2 receptor and interleukin-7 are associated with multiple sclerosis and other autoimmune diseases. So this shows the importance of genes, including HLA genes and
other genes, PTPN22, NOP2, and interleukin-2 and 7 receptor. This table also shows relation of different genes with different autoimmune diseases. Also, other genes, IRA in thymus, its mutation, or mutation in FAS and FAS ligand. This, you remember, FAS is death receptor for in peripheral type of apoptosis. So different types of genes can be related with autoimmune diseases. And role of infections, as I told you, infections can cause changing of our molecules and derangement in our tolerance. Another mechanism of infection to cause autoimmune diseases called molecular mimicry. What does it mean, mimicry? It means that they are similar to our cell molecules. For example, in acute rheumatic fever that is due to some species of Streptococcus pneumonia, pharyngitis, these streptococci have some cell wall proteins that structurally are similar to some molecules in our heart muscle cells. So this is called molecular mimicry. So the structure is the same, is very similar, and the antibodies that are produced against the streptococcal cell wall proteins can also react with our heart muscle cells. That's the basis of rheumatic fever, that it causes myocarditis in patients that are infected with streptococcal pneumonia. So this is a type of autoimmune disease, and its mechanism of action is molecular mimicry. So an infection can cause autoimmune disease by, for example, this having similar molecular structure. After this, Robbie's textbooks start explaining some diseases, important systemic autoimmune diseases such as SLE, rheumatoid arthritis, that we don't do it. Then if we do systemic pathology, we can mention some of the features of these diseases. After this, we start the immunodeficiency diseases. Also, it explains rejection, that if we have time, I will mention some of the mechanisms of rejection that includes direct and indirect pathway. We have little time, just in 5 minutes. I will start immunodeficiency syndrome, and next time I will continue after the quiz. Immunodeficiency syndromes include primary or congenital and secondary types, or acquired. Secondary types of immunodeficiency can be due to malnutrition, infection, irradiation, some autoimmune diseases such as diabetes mellitus. The most important of which is AIDS, acquired immunodeficiency syndrome due to human immunodeficiency virus. It will be for next time. But the primary or congenital immunodeficiency, which is less common than secondary, it includes deficiencies in innate immunity and acquired immunity. In the chapter of inflammation, we mentioned some of the diseases such as leukocyte adhesion deficiency 1 and 2, that they are deficient in some adhesion molecules such as selectins and integrins. So these are included in primary immunodeficiency diseases. Or Chiari-Gaji syndrome that has a problem with phagocytosis. Phagolysosome is not produced. And some complement deficiencies such as C2 and C4, that C2 is the most common. Defects in leukocyte function and maturation, and some diseases such as severe combined immunodeficiency, X-linked A-gamma globulinemia or Bruton disease. These patients don't have any immunoglobulin. Their B cell function is disordered, their T cell function is normal, they don't have any immunoglobulin. Or patients with DiGeorge syndrome or thymic hypoplasia, they don't have T cell function, but their B cell function is normal. So next time I will complete this Bruton and hypoplasia, DiGeorge or thymic hypoplasia, and for example, hyper-IgM syndrome only have immunoglobulin M and not the other classes of immunoglobulin. And isolated IgA deficiency, when these people have the deficiency of IgA, which is the mucosal type of immunodeficiency, they have infections such as infections with Giardia lamblia. And then some systemic diseases such as Wiskott-Ulrich syndrome and ataxia telangiectasia. After this, it starts AIDS. So we will try to finish this chapter next time.